afternoon, everyone. Thank you for the opportunity to speak today, and I'll just speak for a few brief moments for some high-level context around how GRIN Therapeutics was formed, and then I'll hand it over to our Chief Medical Officer, Dr. Michael Panzara, for the meat of the conversation. We're aiming to cover today who we are as GRIN Therapeutics, our current journey to date, and our goal of developing a targeted disease-specific therapy for GRIN-related neurodevelopmental disorder. We'll talk about our clinical programs and how we make sense of them in a targeted way for this patient population. And we'll talk a bit about our involvement with the community and how we can continue that going forward as we head into phase 3 studies. A bit of background on GRIN Therapeutics. We're a New York City-based biotech company. We've been around since 2021. I'll take you through that timeline in just a moment. And we are funded by a single investor to do the work we do, who's fully committed to delivering a novel therapy to patients and families. We were fortunate enough over the course of the last 5 months or so to collaborate with Angelini Pharma, who's also a co-sponsor of this event and whose leadership is in the room today, and quite grateful for their engagement and our partnership as we advance Radiprodil in clinical trials. We've been inspired by the GRIN community since day one. All the work that we've done to design our studies and to execute on our studies has been done in partnership, close partnership with the GRIN community. All patient advocacy organizations have been a critical part to the work we've done and have contributed immensely to every aspect of our program. As mentioned, we're advancing a targeted disease-specific treatment for GRIN-related neurodevelopmental disorder. We like to say that we're targeting the underlying disease biology, which is critical to the work that we do, aiming to go after the cause of the disease as opposed to carving out symptoms that are seen with the disease. Our goal is to treat all symptoms related to GRIN-related NDD. Understanding the broader community in GRIN-NDD is critical. Right now our program focuses on gain-of-function patients, but we realize that there's unmet need across the whole population. And the work that we are doing away from our most advanced clinical trial in gain-of-function patients considers all patients. We consider it from a natural history perspective, which is a key element to any drug development program for rare disease. That's on our list of activities for this year, and we'll be kicking that off. And we're also interested in thinking about the totality of the patient population. That includes loss-of-function patients as well. More on that to come. And then finally, the community is key to everything we do, as I mentioned before. And we'll give you a little bit of a snapshot as to how that has played out over the last handful of years. This all began with the GRIN conference held at Emory University back in 2019, in the earliest days of organizing around this disease process, both as a company, but also as a patient advocacy community. And that motivation, energy coming out of that conference is what led to the creation of GRIN Therapeutics in 2021. We began building that company at that point in time around a molecule called Radiprodil, which is a negative allosteric modulator against the glu-N2B subunit of the NMDA receptor. And from there, haven't looked back. You can see that in 2022, we engaged heavily with the patient advocacy community to design the Honeycomb study. The Honeycomb study was our phase 1b study, looking at patients with GRIN-related NDD with gain-of-function variants. And it was, as you'll see in a couple of boxes later, a successful study, and it's what gave way to our efforts in phase 3. In 2023, we dosed our first patient in the Honeycomb study, and were able to present data over a year ago highlighting the impact of Radiprodil on these patients. What was really key when we chose to engage in this exercise, and you've heard a lot about it today, is to characterize the patients as adequately as we possibly can. We did that in collaboration with a number of different world-leading experts, both clinicians and scientists, to understand how to standardize the approach to characterizing variants. It's an emerging process. There are lots of great ideas. The goal in developing a drug is to have those ideas shared by a number of different clinicians and to publish on it. So we had a very key peer-reviewed publication that came out in the middle of 2023, which talked about the methods by which we characterize these patients. And that's critical to the work that we've done in an effort, again, to standardize the patient population. We were able to deliver data in 2024 in the Honeycomb study showing an 86% median seizure frequency decrease in the patients studied, open label study, 15 patients of data. And because we target the underlying disease process, we were also excited to talk about the impact of Radiprodil on non-seizure symptoms. The goal, of course, is that if you treat the cause of the disease, you have an opportunity to treat the whole disease. And that's something we were really happy to be able to report at ILAE in September of 2024. The caregiver surveys that we've conducted have given us a sense of what you all experience in treating your children. And it's been critical to how we've formulated our views on the phase 3 study, which Mike will talk about in just a moment. But what we really want to drive home is that your input is critical to how we think about developing the drug. And we've been able to get regulators, international regulators, to embrace that view, and can't thank you enough for all of your engagement on that topic. And then finally, we're getting ready to kick off our phase 3 study, what we're calling the Beeline study, and that study will be a phase 3 study, global phase 3 study. We're looking forward to initiating that in the coming weeks. And we'll continue to advance the ball down the field, to use a football analogy, and we'll do that not just in our phase 3 study, but in a natural history study as well. And then again, thinking even beyond that to the broader patient population, including loss of function patients. And with that, it's my pleasure to introduce an incredibly talented CMO, Dr. Michael Panzara, who's been leading the charge behind all the clinical work.
Thank you, Bruce, and thank you to the organizers of the meeting. It's always a great pleasure to be here, and this is my third since joining GRIN Therapeutics in 2022, and again, it's always a great pleasure to be here to meet families, the researchers. It's a great meeting, so again, thank you for the involvement. So I'm going to talk a little bit about Radiprodil, starting off with this concept, which you've heard a lot about, gain and loss of function. We've been using for a few years now this concept of the NMDA receptor having this function of regulating flow through the receptors like a gate, and the chickens running in and out of the barnyard, where you have maybe a broken latch and there's too many chickens escaping, and a sticky latch, and then they all get stuck in there. And we've been doing that for a few years now, but the reality, as you've been hearing, is a little bit more complicated than that. There's a spectrum here. There's a spectrum of what is loss, gain, reduced, overactivity. It's an evolving area that we've now been paying more attention to. And as we think about this, and you think about how this very complex system works, it's certainly much more complicated than just open and close, or too much open, too much close. It's just this balance, because in this complex brain, you have this push-pull of excitatory and inhibitory nerve cells, and those have NMDA receptors on them. So overactivity on an inhibitory neuron reduces function, underactivity on an excitatory neuron is underfunction. It's more complicated than just they go up or they go down. It's trying to establish this balance. So this disruption of the balance from variance is what you all see, and it's these unstable networks that then lead to the symptoms that you all know too well, both from the increased positive effects, meaning excitatory effects that you see as seizures and outbursts, and other inhibitory effects, like neurodevelopmental signs and symptoms. So it is more complex, but if we waited to sort it all out, we'd have no treatment. So we have to try and move forward the best we can with the biology we understand, and again, you're hearing a lot about that, and this is where Radiprodil comes in. So Radiprodil, as Bruce alluded to, is called a negative allosteric modulator. So what does that mean? It means that it causes this change in the function of the receptor, where it binds outside of the pore. So you have, as you see here, just like those chickens, that flow of calcium through the pore, in and out of the pore, And Radiprodil binds outside the pore to the glu-N2B subunit with the goal of restoring some balance to the system. And we're going for the gain of function by one category, so your very gain of function population, because at least in vitro and some of the animal studies, Radiprodil seems to reduce that flow and try to restore that balance. But Radiprodil has also other effects on the NMDA receptor and other parts that will also change this excitatory inhibitory balance, and which is why, as Bruce alluded to, we're starting to expand our target to get more towards the broader population, which I'll talk about more in a moment. And as you know, there's no disease-specific treatments at all for GRIN in the gain-of-function setting, certainly, and we're trying to do that here. So what are we doing in GRIN-related neurodevelopmental disorder? Well, you've already know about our first study, which is the honeycomb study. There was an open-label study. It involved patients with not just seizures, which is typically done. Seizures are typically done in studies like this because you can measure them quickly. They go down or they go up. And there's something, especially in GRIN patients that have so much activity. So it's often done that. But we also included people without seizures to try and see if we can influence neurodevelopment and behavior. We were actually quite honestly surprised with what we saw. An 86% reduction in seizure frequency was beyond our expectations. It's probably twice of what we were expecting to see. So it was a pretty remarkable observation. Most patients actually had a 50% or more reduction, so very robust effect. And you had 6 of the 7 patients were seizure-free 80% of the days during the study, which is, again, a pretty striking result. Interestingly, although in a small number of patients with all the caveats that happen there, we also saw that both clinicians and caregivers saw a global improvement in all patients, whether they had seizures or without seizures. So we started to see reports of behaviors across the board improving and using these caregiver and clinician global impressions of change. That prompted us to do something which is highly unusual in this space, which is to go to the global regulatory authorities and turn a phase 1 study into a phase 3 study, so we didn't waste any time, and they agreed. In fact, we got all these designations: rare pediatric designation, breakthrough designation, orphan designation, prime. These are all the different ways that the authorities, both EMA and FDA, sort of acknowledge that this could be something different here, and we're going to help you move this forward as quickly as possible. And that's basically what has gotten us to where we are today. The open label study continues. We quarterly have that reviewed by our Data Safety Monitoring Board, and the safety is essentially unchanged. And actually, the last review of the safety data was actually about 2 weeks ago, so it's actually consistent. It says June there, but it was about 2 weeks ago, and it's actually consistent with what we have been seeing. So what happens now? So the Beeline trial is the phase 3 amended phase 1 study. So essentially, patients who are in the study now are not shown here, but those patients are going to continue on Radiprodil at the dose they were receiving, and we're going to continue to collect long-term safety and efficacy in those people. So those people don't have to worry about placebo. Those people will stay in that trial, and we will continue to provide the drug up until availability, hopefully, if it eventually works. We actually now, in the global phase 3 amended study, we're going to follow a similar pattern to what we did in the honeycomb study, where we're going to have patients with at least 4 countable motor seizures, or patients who have less than that, randomized to 2 groups with either active or placebo, looking at whether we have an effect on seizure reduction in a placebo-controlled way, because that is the gold standard here, is to establish an effect versus placebo, or patients without the right number of seizures having an effect on the behaviors and some of the more severe non-seizure symptoms associated with the disease. We're also expanding the age range from 1-month-old to 18 years, trying to get as broad a population as possible. And essentially the gain of function population per the publication in 2023 is part of the inclusion criteria. The one difference between the cohorts, other than the amount of seizure activity, is also how long the placebo-controlled period is. The placebo-controlled period in the non-seizure cohort is 24 weeks, and that's really so we can hopefully see changes. The issue with assessing behavior and neurodevelopment is that the tools we have are pretty primitive. They're insensitive, meaning when you have an insensitive measure, you need to have either a lot of patients or follow people for a long period of time. And unfortunately, we don't have a lot of patients, so we have to follow them a little longer to see if there's a change. Otherwise, we might not see any change at all, and we won't know. So that's the rationale for having a 24-week placebo-controlled portion in the non-seizure cohort. But at the end of all of this, all patients will get open-label Radiprodil, and that's going to be provided up until commercialization, which means the market access, meaning reimbursement by the countries, depending on the relevant country. But everybody gets the drug, and we'll continue to follow both for safety and efficacy. This is more of the patient populations involved. I already talked about this. Importantly, that efficacy that I showed you for the honeycomb study, that was on top of on patients' existing anti-seizure medications. Patients actually came into that study with an average of 28 seizures in a month, and on an average of three or so anti-seizure medication. So very, very intractable seizure patient population. So we're still looking for those who had an inadequate response to at least 2, but and they can come in with as many as up to 4 anti-seizure medications on which Radiprodil is added on top. So we've been encouraged that by adding on top, drugs seem to be quite well tolerated. And then the hope will be, once we get through this placebo-controlled period and we follow people for longer periods of time, obviously the ultimate goal is to start to wean off other drugs, but not for the actual study itself. And again, there's some other exclusion criteria. There's, as usual, a long list of these things, and if folks are interested, we'll eventually be putting more information on the website and connecting interested families with our investigators. So one important part of this program is this concept of measuring non-seizure symptoms. And this is, as I mentioned, has been a challenge. There's a lot of tools out there that are done, but none of these are really good. So we actually went to the community and did a survey to figure out what exactly are the most meaningful things to measure. Because a lot of these tools measure a lot of needless things, and they take a lot of time. And those of you who've been as part of these assessments, some of these neuropsychological tests take a lot of time. They're very tiring. They're very disturbing, really hard on families and the patients themselves. So we went to the community and we said, okay, let's see if we can develop our own tool to measure what matters to GRIN families. And so we did a survey. And this survey was followed up then by interviews with families and caregivers, as well as interviews with advocates around the world. So the survey was done in North America, but we took the results to advocates around the world to see if what we learned in North America applied. And then we did what's called a developing of a conceptual model. It's just a fancy way of saying develop almost like a diagram of the most important things to the community. And then what we did is distill that down through methods, psychometric methods that are done by experts in this field. And they basically bucketed out into these 5 categories on the bottom, which I'm sure sound very familiar, as the things that if we could have an impact on these things, this would be a meaningful effect on the population. So the focus here is trying to improve these 5 things. I don't know if you can all see them: receptive and expressive communication. Obviously, communication is life-changing. That's what helps us interact with our environment. Gross and fine motor, meaning big movements and finer movements of the hands. And then, of course, this dysregulation or this difficult behaviors and trying to bring those types of things under control. It was felt that these were going to be the most impactful ways of hopefully helping patients and families. So that is an important endpoint. It's an important endpoint across. This shows the other endpoints. Generally, seizures will be the primary endpoint in the overall study, because that's, like as I said, something you can measure. It's something that can be in 12 weeks. But these additional endpoints around the CGI, this GRIN-specific CGI, as well as other tools to try and not just understand, which seems to be the common thing to do, is can you reduce seizures? It's can you change the overall course of the disease? That's the objective, and lofty goal, but we'll see what we can do. This is where it's being done. It's being done all over the world. You can see the dots across Europe and North America, Australia, Asia. The goal will be to try to collect enough patients across these regions to show that we have a consistent effect across different ethnicities and patient populations. So I've already alluded this, as did Bruce, a few other things. The top here is our B-line study. We have to think about, okay, so now that we have this promising effect in honeycomb, and we have this evolving understanding of the biology, where do we go from here? So you have the B-line study, as we mentioned. I just described all of that. That's the classic, here's how you define gain of function. Let's see if it works. But one of the things we've been challenged by is there is limited natural history data. You've heard that several times today, actually, that natural history data is critical to collect. So because we have to understand the disease in order to best treat it. And so we are funding our own natural history study in North America, a GRIN study, by GRIN Therapeutics, I should say, in North America, and that should be, by the end of the year, that should be fully underway. And you already heard about the EuroGreen study today. We are working to support that study, because we wanted to make sure that we made sure we collect regional information outside of North America. And the team, EuroGreen, has been fantastic to work with, because we are trying to align the two studies as closely as possible so that we can integrate the data and best inform the community. And then finally, I alluded to this a few times also, it's like, where else do you go where NMDA biology is relevant? And so we already have gone there with a study called the Astroscape study in tuberous sclerosis complex and focal cortical dysplasia. These diseases have an overrepresentation of the LUN2B NMDA receptor, which leads to some of the problems of the disease, both seizure and non-seizure. We're seeing if this mechanism could be applicable to those patients. Other GRIN neurodevelopmental disorder studies, we're actually moving into now. This, as we said, there's a spectrum from gain to loss. We're now beginning to move through indeterminate patients and loss of function or reduced function patients to see if we can have a biological effect in those populations, given some of the biology we've learned. And other indications, it has been really exciting to have spoken to many researchers here and in other meetings where this GLUN2B biology is important in so many other rare neurological diseases, neurodevelopmental disorders, and we are now exploring moving into some of those also, either on a research basis or even a clinical basis. So, you know, once you have something that could be biologically meaningful, the goal is to get it out in as many places as possible to see what to make the maximal impact. So in summary, this is essentially what I said. Since I'm running out of time, I think I don't see a clock or anything, but I'm sure I'm out of time. This is sort of, Radiprodil, we're bringing it forward. We have our phase 3 study ongoing. The CGI, as I've already said, we're hoping once this is validated in our study, because we're validating it during the study, it will be provided to the community for use in other ways. So that's going to be out there and usable by anybody who wants to do it. These natural history studies, also those will be data that will be shared with the community. And we're looking at these different ways of treating the disease. Some ways that you all can get involved, and I think you've heard this throughout the meeting, and you're going to continue to hear it throughout the meeting. Staying connected, knowing about your situation and what might be the best. You heard other treatment trials today. You've heard about what we're doing. It's important to know, the more information you have, the better empowered you are to make the choices for you and your families. There's options out there. We're one. And it's really important just to take control of it and learn as much as you can. And then some of you will come to us, and that's wonderful. And you can always find out what we're doing at GRIN Therapeutics. Those who haven't been to the GRIN portal, another way to find a lot about your disease, take a look there. And just thank you. Thank you for the opportunity to speak. We have collaborated with just about everybody in here, or we have been open to collaborating with just about everybody in here, which has been fantastic, and I'm grateful for that. The participants in our trials, this is another talk that I give about how hard it is to be in a clinical trial. Some people have heard this talk and why it's so hard to be in a clinical trial. That's for another day, but it's hard to be in a clinical trial. So participants who do that are special people and their families. It's very difficult. And so thank you for, since 2019, being the inspiration. That's all. Thank you.